Hello and welcome to this webinar titled Manufacturing Strategies for Biosimilars, a Case of Continuous Capture, hosted by Biopharma Asia magazine and presented by Solomon Alva, Antibody Purification Group Lead, Senior Scientific Manager at Biocon Research Limited, and Yvan Ruland, PhD, Technology Director, Asia Pacific Operations at Novasep Asia. My name is Ursula Balog and I will be the moderator. Now, please allow me to introduce our first presenter, Solomon Alva, Antibody Purification Group Lead, Senior Scientific Manager at Biocon Research Limited. Solomon Alva is a Senior Manager at Biocon, leading the Antibody Purification Laboratory. He has over 12 years of experience in biologics development. His area of expertise is bioseparation technologies and life cycle management of product development from early stages to commercial manufacturing. He has been instrumental in developing the purification platform approach and process for biosimilar mAbs at Biocon. Solomon has a proven expertise in alliance management, leading multiple cross-functional initiatives with co-development partners and steering academic industry collaboration projects for Biocon. He leads a group of 25+ team members with PhDs and postgraduate degrees in engineering and science focused on development and scale-up of purification processes for novel and biosimilar biologics. Solomon has completed an Executive General Management from Indian Institute of Management, Bangalore, and is a Master's in Science from Bangalore University. Welcome, Solomon.
Thank you, Ursula, for that wonderful introduction. So I'm going to jump into my slides. So essentially, what I'm going to do today is run us through a case of continuous capture. And I'm going to keep in mind, as a biosimilar manufacturer, our approach towards using and sort of adopting the continuous capture platform. So I've put up a slide over here, and I think it sort of sums up where a lot of thought process right now in the industry is in moving towards continuous capture and continuous processing. And in this case, basically, I've put up a slide of end-to-end continuous processing. For a biosimilar manufacturer, the idea is this is a place where the industry wants to move, and so does a biosimilar manufacturer want to move. But in that journey, there are a number of points to consider. One essentially is the high level of automation and control expected when one moves to a continuous manufacturing process. There are a lot of enabling technologies critical to the success of a continuous process. For instance, the PAT is required during the multiple testings required for the smaller lot sizes and multiple lots during a manufacturing batch. Approaches are critical to the development of a continuous process. So viral validation approaches, process characterization approaches would be critical to be modified in comparison to a batch chromatography. In addition, there's a large volume of data which comes out of a continuous process, and this needs to be processed, analyzed, and continuously monitored. And this heavy amount of data, tools such as MVDA, enabling tools such as MVDA are available, but the thought is that multiple additional tools may be required to be able to manage a continuous process. One of the promising features, of course, in continuous process is, and this innovative landscape in which the industry is moving towards, is that the health agency, the FDA, has come out very supportive of continuous processing, starting with pharma and now talks going on with the emerging technology groups that they have set up to engage with the industry, move towards continuous process, to enable and help them to move towards continuous process. So there's a lot of supportive environment of using this innovative technology. And this is where sort of Biocon comes in. Our goal, essentially, our vision of the company is to use innovative technologies enabled to get affordable biopharmaceuticals for patients and partners in healthcare systems across the globe. And one of our endeavors has been to evaluate continuous processing so that we could bring down costs, so it makes drugs more accessible for patients across the globe. And Biocon essentially has managed this by developing the vertical format, where we essentially have biologics, a core focus right now in the medium and long-term vision of the company, and in the long term, the novel biologics. And Biocon has the demonstrated ability and demonstrated over the last few years with the launch of trastuzumab and the approval in the US FDA. And in addition to this, insulin glargine, which was approved and launched in Japan, and a recent approval in the EU too for glargine and pegfilgrastim in the US FDA. So a demonstrated of capabilities for Biocon in the biosimilar space. So essentially, in this talk, I'm going to focus on the continuous capture, the multicolon chromatography, run through a case study of our process development approach, move on to the results of that approach, and sort of conclude on how as a biosimilar manufacturer, we are observing this to be a unique platform which can enable the process to handle high titers and various upstream processes such as fusion or the quasi-fed batch processes. But first, trying to get an understanding of why the proteinase step turned out to be the right step to sort of take a first step into moving into continuous process has definitely been the high resin cost of a protein A resin, and the capacities which are limited to around 40 to 60 gram per liter results in large resin requirements at a manufacturing scale, resulting in high buffer consumptions too. But from a unit operation point of view, it's clear that this step's key requirement is capturing the product and concentrating by around 5 to 10 folds and reducing host cell contaminants. But in general, lesser influence on the product quality has been observed by multiple manufacturers, including ourselves, essentially. In this slide, essentially, what I'll do is I'll sort of introduce the continuous capture to some of us who are observing it for the first time. And essentially, the process is different from a batch process, where in a batch process, it's clear that the chromatography resin is not completely utilized during the loading of the product onto the column, where you typically have anywhere between 50 to 60% of the resin utilized if used optimally. And in cases of commercial manufacturing, at times, depending on variabilities in the titer, that utilization dropped down to as low as 40%. What happens in an Smcc process, and I'll short while I'll show you a small animation of how it works. Essentially, you have multiple columns which are operated independent of each other, and therefore you have the ability of allowing each column to be in different phases of the unit operation. Here's a small animation to sort of take us through how this happens. So you have a column which was completely loaded, which now can move into a phase of elution, whereas the other columns move on to go into the primary loading phase. Now, a key thing to notice over here is you'll see that there are two columns in loading. The first, where you have a column which is maximally utilized, and the breakthrough of the first column is captured onto a second column, which is known as the second pass column. And this second pass column captures all the breakthrough of the first column. And this is, of course, one of the areas of development. Decide at what time or at what column volume one would want to make the switch for the second pass column. So this is essentially an animation of the sequential multicolon chromatography. So what are the benefits and what are the drivers to move towards this? Definitely, if as a biosimilar manufacturer, you're looking at reducing footprint when you're designing a facility or in your existing facility, it's an opportunity to reduce the column diameter. A second driver could be the need to maximally utilize resin usage and thus have an opportunity to save on COGS. And in the process, one essentially has a reduction in the buffer consumption, potential to even reduce the tags in your manufacturing facility through disposables, and other side opportunities of reducing the load on the inventory management of handling smaller quantities of protein A resin. So in this slide, essentially, I'll just quickly run through it, but it's a snapshot of other considerations for a continuous capture. One consideration is and I'll talk more about this. A quick way to assess whether one has an opportunity to really gain from the continuous capture step is quickly evaluate one's DBCs and SBCs for the protein A of resins at hand, and I'll talk more about this in a later slide. Another consideration one can keep in mind is that there are different types of MABs in the portfolio, whether it's IgGs or fusion MABs, and make an observation of the differences in their capacities. And as you can see in this data, we've seen differences, significant differences in the fusion MAB and the IgGs, the capacities of these. Another consideration, of course, is the stability of the product in the harvest and the clarified harvest. And this is a critical consideration because this is one of the key features of the multicolon chromatography, where there's an opportunity to connect the multicolon chromatography with perfusion cultures and thus minimize the hold time of the product and thus the degradation rate of the product in the cell culture harvest. We'll talk a little bit more about this in the later slides. So I'm going to go into a development approach mode, and this is where I'll start sort of diverging from typical presentations on continuous capture, and we're looking at this from the lens of a biosimilar manufacturer. And the consideration for other biosimilar manufacturers is how does this technology, when adopted, allow to be utilized in a platform approach? And it really starts with development. When we're looking at development, typically at biosimilar manufacturers, we get anywhere between a year to under a year sometimes to develop a process, and it's critical at this juncture to have a platform process. So essentially, I'll run through a development approach which is well established and share some of the data that we've got from the development approach, and then move on to a slide which then talks about the results of the case study. So it's critical to be able to evaluate the capacities of the resins at various residence times. And this is the data which is presented over here, where we've done dynamic binding capacities for a chosen resin. And this enabled us to get the required model inputs so that we could use it in the process model, which is the linear driving force model. And we essentially obtained the Qmax and the mass transfer coefficients, which were then used to simulate the operating binding capacity curves at different residence times, which is the second graph, which then enabled us to simulate and make a decision on whether we wanted to get optimum utilization of the resin or optimum utilization of productivity. And here I'll take a pause and quickly explain our thought process on what options are available, and one can use a solver which is available in Microsoft Excel, something we did, or you could interact with the many bioservice providers who provide their own models to be able to optimize for one's objectives at manufacturing scale. Now, essentially, this optimization tool enables us to determine the optimal residence time, number of columns, and configurations to then determine whether we want to move into minimizing the resin usage or maximally increasing productivity, or a balanced scenario where one would want to shrink the footprint and allow maximum product to be produced in a given time. So that's what we do essentially in a solver-based optimization. And our experience has been in our scenario, we use the balanced scenario approach to move in the direction of balancing out the resin usage and the productivity. Finally, at the development stage, it's critical to perform and validate the model. And once you validate the model, you can always go back to the model and change scenarios for various molecules. So once it's established that the model works, it gives confidence to accelerate development for the other biosimilar portfolios. And this is a key point to keep in mind when one is looking to accelerate timelines. So in this slide, and I'll quickly run through it, essentially gives an impression that when you're overloading a column, one would, and this is something that when we started off a few years back, we were curious to see the changes in the contaminant profile when overloading a column, and we had seen with its impact on viruses, which I'll talk about a little later. But it was clear that overloading the column and allowing product to be captured in a second pass did not impact the clearance in comparison to a batch for 3 mAbs in this case, where you can see that the host cell proteins and host cell DNA are comparable to the batch process. And even in the case of the leach protein A, in one case we did see slightly elevated levels in the case of the first mAb, but it was well within the batch chromatography. And it will be important to look at some of these parameters when you're validating the model and running your runs. And one approach that we took is we decided to run a continuous run for the 8 to 16 hours to test the system and test the process and ensure that other parameters are taken into consideration. So essentially, there are 2 modes, and this is another thought process for a biosimilar manufacturer, where we essentially are looking to lock our process before we even scale up. And so therefore, the first clinical material generation for either the phase 1 or the pivotal study is a locked process. So when we're doing a novel antibody or a novel product, there is an opportunity to make changes between your phase 1 material generation and your commercial process. In our case, we decided to lock the process earlier on. So this case study is a demonstration of the media savings that one can get if one were deciding to have a marginally different process between your clinical and your commercial scale. And in this case, you can clearly see we've used a scenario where we've looked for maximal resin usage with a resin volume coming down from 57 liters to 16 liters, and you can see a huge savings of $500,000, where there's a return on investment of scaling up just 4 monoclonal antibodies. The scenario that we really went forward with is not the clinical media savings. We went forward with commercial media savings, and the key reason for us over here was we were looking at locking our process early on during the first scale up, so we were able to minimize the amount of comparability that needed to be done in later stages of the program. So a quick back of the hand calculation one can do. In this case, we've used data, but essentially, if there is a dynamic binding capacity, and we've shown some of the DBCs we've had for different mAbs and fusion mAbs, and the calculated static capacity, one can take 70 to 80%, and maybe upwards up to 90% of operating binding capacity of that static capacity. And one can easily quickly calculate the commercial media savings. And in our case, of course, this is the actual case study data, but it was evident for us that we were looking at the commercial process, and for us, media savings of between 20 to 50% in the case of the fusion map was critical for us to consider this as a platform, essentially. On this slide, I've talked about a sort of a forward-looking way of looking at and how we're looking at the continuous capture step, where we've evaluated various levers for increasing productivity. One of the levers, of course, is the various intensifications which are possible within the existing batch process, and I've termed that as modified batch or optimized batch. You can see there's some gains in the productivity. The second lever, of course, is the various technologies available from the various bioservice providers. In our case, we evaluated them in two different modes. We evaluated them when the titers were in a 1x mode. You can see that in the first set of data, the productivity being 521. And we also evaluated it at higher titers. In this case, I've put it as a 2x technology 1, technology 2, and technology 3. So it's clear that at 2x titers, there's no difference in the productivities between technologies. But essentially, a note on technologies, and our experience has been the key consideration over here is based on the manufacturer's requirement and manufacturing scale of the ability to handle, let's say, multiple columns or multiple configurations. And one would then look to different manufacturers to suit one's needs. In our case, it was very clear that the technology did not play a role in our consideration, and we would take scale-up parameters more into consideration. The third section of this graph talks about the opportunity to use the inline concentrator for determining if one can get higher titers in the load of in the capture step, essentially. So in this case, we used an inline concentrator, and one can see dramatic benefits in terms of productivity using an inline concentrator. One will need to keep in mind that the inline concentrator has this advantage because a typical loading scenario for a protein A step is around 20 to 22 series of loading, which is at least 50% of the operating phase of a unit operation. So reducing this volume using an inline concentrator, you can see, has significant benefits on increasing productivity. A third avenue, of course, a third lever to evaluate to increase productivity in the continuous capture is looking at the latest protein A resins which have been launched over the last 3 to 5 years after the expiry of the patents. And the portfolio of protein A resins we evaluated along with our control resin. We did not see significant gains in productivity, but recent data, which is not on the slide, has indicated there are potential resins out there being launched which allow for both maximally utilizing the resin and getting high capacity at lower residence times. So in summary, there are multiple levers for increasing productivity, and this is something that can be done if one has to meet the objective of COGS reduction or constricting one's capacity at manufacturing scale. So I'm sort of moving to the end of this presentation, and I thought I'd talk about a few challenges which are currently viewed in the industry, each of which there's tremendous amount of references and literature out there. For instance, in the viral validation, Sarah Johnson and Kurt Brosen last year published their view on the viral clearance approach for a continuous capture and for a continuous processing, for that matter. And our approach at Biocon for the viral clearance has been in line with the opinions of these FDA representatives. And one of our thoughts in this regard has been we're looking at overloading batch chromatography resins during scouting studies early in development to be able to assess if the viral clearance is compromised while overloading or capturing a second pass in a multicolon chromatography approach. So that's the thought on viral validation. There's ample amount of information out there, and a similar thought process for pooling and lot sizing, which has been belabored in the industry, and with the entry of FDA giving the opinions on this front, very clear guidances out there on rationalizing the ability of defining a batch in the case of continuous capture. Other enabling technologies, and I'll talk a little bit about this path and integration with cell culture on the next slide. But in general, a continuous capture can be an early, quick start for a manufacturer looking to move towards continuous, considering that the tools required for a capture step are not of the magnitude of evaluating product quality attributes, but more so for evaluating protein concentrations, column volumes, pH, conductivity, and if the case is required, the host cell contaminants. So the requirement of enabling technologies are quite low when it comes to PATs for a continuous capture step. One of the key things, and I'll talk a little bit on the pressure drop, this is a critical factor during scale-up, especially if one were connecting columns of upwards of 2 columns to 3 columns at the manufacturing scale. One will have to keep in mind, if they're connected in series, the impact on the back pressure is something that one needs to evaluate in a pilot scale before moving on to manufacturing scale. And this is where a careful consideration of the bed height and the flow rate is important. Either it can be put into the models earlier on, or one needs to evaluate this at a pilot scale mode before moving into manufacturing scale. And I'll talk a little bit, and I mentioned I'll talk a little bit about the integration with Upstream Profusion, and this is a critical point, especially for manufacturers. In our case, there's always the opportunity to move from the batch mode to a profusion mode, and if one were to adopt a continuous capture technology, what would be the parameters which one would have to take into mind while developing this technology? There are a few parameters one will need to take into consideration. Definitely the perfusion rate would have to be synced with the flow rates of the continuous capture step, and thus we'll be able to get a seamless continuous process from the upstream right down to the low pH step. Another important consideration is the opportunity to consider a dynamic control of the capture step because of variations in the titers which one could expect in a continuous cell culture. While this might be an option even in the batch, fed batch culture, it is an important consideration when considering a perfusion culture where there are titer variations when the upstream process has not hit the steady state. And therefore, there are manufacturers and, we're working on an in-house capability to be able to have a dynamic control for the multicolon continuous capture. And I would like to sort of summarize at least the approach from a biosimilar perspective. I think one of the key advantages that we see here at Biocon for the continuous capture step is this opportunity of considering this as a platform approach, and essentially the options of using this technology for bed batch or a perfusion, depending on the capabilities one has in the manufacturing side. And in our case, it was important to develop a technology or enable a technology which allowed the ability to capture product from varying titers, varying technologies in the upstream. And as I mentioned earlier, a lot of favorable ecosystems have come into place in the last many years, and has really been a driver at least for us, to think about continuous capture and enabling continuous processes, with both bioservice companies providing enabling solutions and the US FDA very supportive of continuous processing. So, in conclusion, the MCC step provides significant benefits for commercial and clinical media savings and significant buffer reduction and the enablement of using ILC to increase productivity, and a very promising continuous capture from our case study, essentially. I'd like to, very quickly thank the people who developed this data, developed the process, and sort of been steering this effort at Biocon. Sandeep Hatpe has led a team of two, for Vijay Manohar and Yvan, who contributed immensely to this, and to my mentors, Jyoti, Partha, Vijay, and Anuj, for helping me to get this opportunity, and definitely the team at MDL, who've always been a constant support. Thank you. Thank you, Ursula.
Thank you, Solomon. Once again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation. Please feel free to send in your questions at any time during the presentation via the question tab located at the right-hand side of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second presenter, Yvan Ruland, PhD, Technology Director, Asia Pacific Operations at Novasep Asia. Yvan heads the technical operations of Novasep Process Solutions in Asia Pacific area. He provides expertise in purification technologies and drives the process development projects for Asian customers in biopharma and pharma industries. He has over 15 years of international experience in Europe, US, and Asia in developing chromatography batch and continuous processes for downstream purification of biologicals and active pharmaceutical ingredients. Besides being a graduate from University Paul Sabatier in Toulouse, Yvan has a PhD in organic chemistry applied to biomolecules. Welcome, Yvan.
Thank you, Ophelia. So, in this presentation, we are going to discuss more in details about the continuous chromatography step itself. As a matter of fact, the concept of continuous capture can sometimes be a little bit confusing, as there are many different solutions that have been developed by different suppliers and different process development teams at the biopharmas. So what we will present here are the solutions that are available on Novasep equipment. So a few words about Novasep. It's a company provider of services in the field of molecule production and purification for life science and chemical industries, based on a pool of specialized technologies. The business model is based on two sets of offerings. One is process development and contract manufacturing. So Novasep is a CDMO company. But Novasep is also an equipment supplier with services of process engineering and supply of equipment for pharma and biopharma industry. In the field of continuous chromatography, Novasep is actually a pioneer, as the company was created in the 90s to propose to the pharmaceutical industry some continuous multicolon chromatography systems. So this technology was actually already existing, but it was used at industrial scale, mainly in petroleum and chemical industry. So what Novasep did is that it made the technology suitable and available for the separation of APIs of very close chemical structure to purify the API from impurities of very close structure, this in cGMP environments. So those systems were actually operated at high pressure with stationary phases of small particle size. So to develop and optimize continuous processes, deep understanding of the characteristics of the separation is required, and modeling and simulation tools have been developed internally since that time for that purpose. The CDMO, Novasep is using this technology, And we have 3 manufacturing sites that are equipped with some of the largest MCC systems in the world, with 100 tons a year production of APIs. Oops, I think there is something. Okay, sorry, I think the slides were not switched. I think that's the case now. Okay. Sorry about that. So, Novasep had to address some quality questions that are linked to a switch from a batch to a continuous process. The production constraints are actually very different between pharma and biopharma industry. As in pharma applications, there are solvents in the mobile phase, which prevents from the risk of bacterial and viral contamination, which are critical risks in biopharma applications. However, there are some quality challenges that can be approached the same way during process design and process qualification, with some examples listed on this slide, like the batch definition or the pooling strategy, to mention a few of them. So on this slide, this is the system that is being launched for capture or flow-through in proteins applications. So we call it the Biosc pilot. So the one we show on this slide is a standard equipment with a capacity of 5 to 100 liters per hour. This is a modular system that is made of different units that can be connected together. So a single module, the picture on the left, the single module is going to be a typical batch LPLC skid. But the same system, the same skid can be operated under continuous mode with the same unique control PC by adding some modules, one module for one additional column. As soon as we have more than one module, we are starting to talk about multicolon chromatography. So as we mentioned here, Smcc. And this equipment has been designed so that it can provide enough flexibility to handle different processes. So with the same machine, with 4 columns as an example, we can run a 4 columns multicolon capture step and continuous, but under different modes of operation. So we are going to discuss about these different modes here. So they are represented on this slide. The first one on the right side is what we call BMCC, with columns working in parallel and not connected together. So BMCC stands for batch multicolon chromatography. So the columns are not connected together again, and they are undergoing each one a typical batch bind dilute process. By working in parallel, we can adjust the loading steps so that the columns are not loaded at the same time, and we can adjust this so that the system is loaded continuously. So it's going to be a continuous multi-column process, but still using a batch process. The second one we call Smcc, for sequential multicolon chromatography. So this is a historical continuous capture process developed by Novasep, with still a continuous loading, but columns being connected together at some point, so that more protein can be loaded and at a higher speed. So as a consequence, with the possibility to make savings on resin consumption and on buffer consumption. The third process on the left side, so Smcc, MS stands for multi-step, and this mode of operation allows for multi-step chaining, where on a single equipment with a single control system, the columns from the capture step are connected to the next ion exchange column which is connected itself to a third one for the second polishing. So let's start first with more details about each mode of operation. The batch multicolon. So on the next slide, we can see a typical bind-dilute batch process with a load step, then the wash, elution, regeneration, and calibration. And this figure shows a time sequence representation of this process that we also call chronogram. Here on this slide we see the cell process that is applied to two columns using a single pump for loading. So as soon as the loading is over on the first column, which is on the left side in the graph, that's the red part. As soon as the loading is over, the loading is continued to the second column, which is at the right part of this figure. During this time, during the loading of the second column, the first one is processed. And once the second column is loaded, we go back to the first column if it is available for loading. So to do that with two columns, it means that the duration of the non-loading step should be shorter than the duration of a loading step. If this is not the case, we are going to need more than two columns. As the columns are operated in parallel and they are never connected together, this mode does not require any adjustment of the batch recipe. So it represents a simple way to achieve continuous loading without additional process development or validation for the bind-dilute process itself. Nevertheless, there is still a critical difference with the single column batch process, which is linked to the duration between two elution steps that are going to be shorter in the case of batch multicolon. What we can see here is a situation when the feed titer is increasing and the loading time is becoming much shorter than the sum of the other non-loading steps. So when the loading is over on the second column, the first column is not yet available to receive the feed, and we need to add a third column in the system, or even a fourth one, even more depending on the conditions. So if we consider here a cycle time of 2 hours for the batch process, the time between two elution steps is 1 hour 45 minutes with a single column. But in the case of the batch multicolon, is going to be, in this case, only 15 minutes, which is not enough to perform the viral inactivation, as the resistance times at low pH are typically much more than that, around 1 hour. So when switching from a batch process to a batch multicolon process, the validation of the chromatography process itself will not need to be done again because we don't change the recipe. But there are other questions that need to be addressed, like pooling strategy or viral inactivation. Another drawback here is the number of columns that are required to ensure the continuous loading. Here, on the next slide, we are representing the number of columns that are going to be required according to the titer in the harvest. So we can see two curves, the blue one and the orange one. The blue is a simulation that is made with a protein A that would be of low capacity, 18 grams per liter as a loading. The orange one is higher capacity with 35 grams per liter as loading. On the right side, the table, the blue table, is where we have the condition, the parameters for the separation that we have used for this simulation. So we can see that very fast like above 3 grams per liter, in the case of the low loading protein A, we are going to need a very large number of colons, like 5 colons, and even 7 colons above 4.5 grams per liter. For the higher loading protein A, we are already at 4 colons above 3.5 grams per liter concentration. So this slide is about the productivity. As far as productivity is concerned, there is no big interest to switch from a single batch colon to multicolon batch, because the productivity values of the multicolon modes are always slightly below or the same as the batch process. So on this graph, we can see some blue curves that are again for low capacity protein A, with the dashed line are the productivity values according to the titer for the multicolon process, and the solid line is the batch process. So we are just below or same as the batch process for the productivity. So this is due to the fact that in the case of multicolon batch, there are some colons that can be ready for loading at some time, but they are going to be kept a longer time under equilibration because the loading is still going on another column. So as a conclusion for this mode, it can help when we want to apply a continuous loading without changing the batch recipe, but it requires a high number of colons when titers are increasing, and it does not help to increase productivity and does not give advantage in terms of media and buffer consumption compared to batch. So the main advantage is like we can operate continuously a batch process, a batch mode. But if you want to use less colons and make saving on resin consumption, or if you want to reduce the colon size, the system size, keeping a continuous loading, we are going to switch to another process, which is the next one here, that we call Smcc. So this sequential multicolon chromatography process is of the same type of the one described in Solomon presentation, because now the colons are getting connected together at one point during the loading step. So by connecting colons, we can increase the loading amount, or we can increase the loading speed, as the breakthrough coming from the first colon will be captured by the next one, instead of being lost in the waste fraction. So we are still very close to a batch process, with very close to the batch recipe, but the batch recipe still have to be changed, but not drastically. Just a few parameters are changed, like the number of CVs, of course, for loading, because we are loading more, the speed for loading, and the colon height is also a parameter that we have to determine to optimize for this multicolon process. One more comment on this slide is that the Smcc is still a cyclic process. So one cycle is made of several periods. We have the same number of periods as the number of colons, actually. And here on this slide are represented 2 sequences for the periods 2 and 3, where the colons 2 and 3 are being loaded first, and will be ready soon for being processed with the load for the next non-loading steps. We can also represent the cycle using a chronogram. So here on this chronogram, there is no color when the colon is connected with the previous one. So on the left side, this is colon 1, in the middle it's colon 2, and on the right side is colon 3. So at the top, we can see that colon 2, there is no color. It's because this colon is connected with the first colon. 1 and 2 are being loaded together at the top side of the chronogram, and at the same time, colon 3 is being washed, eluted, regenerated. If we go a little bit below, colon 1 is saturated and need to be processed. So column 1 is undergoing the wash and the addition steps, while column 2 is being loaded first and connected with the column 3. So this Smcc process is closer to the other small molecules continuous processes like SMB, because we are connecting the colors together now. So this is making the method a little bit more complex because we have to deal with additional parameters. And to develop such methods, we have to use modeling and simulation tools in order to well understand and characterize the separation. So this is simulation software. I'm going back. That is, this software has been developed for the capture step. So what we can see here are two screenshots. On the left side, this is just the condition of the batch recipe. And on the right side, we are showing the breakthrough curves that have to be previously prepared in order to be able to simulate some process with this software. So on the right side, we can see there are some different colors like blue, red, and green colors, and some points. So the points are experimental values that have been collected at laboratory at different speeds, and one speed for one color here, for one curve. So from this point, a model is created by the software, and the fitting is made with the experimental points in order to validate the model. And once this is done, the software can calculate what would be the total capacity of the media. And here, I don't know if we can see, but on the bottom side and right, for this example, the total capacity is calculated at 37.5 grams per liter. So on the next slide, we see a screenshot of the software where the simulation and the optimization is performed. So we know at this stage the total capacity, and we are going to, so now on the right side of the slide, this has some zoom of the screenshot. So we are going to enter some optimization parameters, some constraints for the optimization. Here we are requesting the software to optimize a process with a number of columns between 3 and 6. The column length must be between 8 and 15, and the loading velocity must be below 350 centimeters per hour, because we know that we have previously made some experiments to check the pressure drop at that speed with different column lengths. The column capacity, the loading capacity is going here between 60 and 90%, and the maximum pressure of 3 bar. So then we can start the simulation, start the optimization, and we get some results in terms of process parameters. So here in this case, the speed is 348 centimeters per hour for the loading. Number of columns is 3, so this is at the top side of the zooms. Columns 8 centimeters, 8. And then we get from this at the bottom some results in terms of performances with the media saving, clinical media saving, or commercial media savings. So commercial media savings are the savings made with the assumption that we are going to use the resin until the end of its lifetime. While clinical media saving, we don't use the resin until the end of its lifetime. So on the right side, we have the productivity gains and the buffer savings. Next slide. So the question of how many columns. On the left side of this slide, we have the graph that we introduced previously with the multicolon batch process. On the right side, this is the simulation made under Smcc process. And we can see that from titers below 3 grams per liter, we need only 2 columns with high capacity media and low capacity media. But the maximum of columns needed here is 3 columns in the case of the low capacity media, even for titers as high as 5 or 6 grams per liter. Next slide about productivity. So here we show some simulation results with what would be the performances and what would be the optimum column configuration, how many columns for different titers, 1.1 gram per liter and 5 grams per liter. In the case of the low capacity protein A with a total capacity of 37.5 gram per liter. So what we can mention here is the fact that for low titer, 1.1 gram per liter, the productivity gains, the buffer and protein A savings are very similar for 2-column process and the 3-column process. Even if the optimum calculated by the software is 3 columns, we can do almost the same, have almost the same performances with only 2 columns. So this is due to the fact that we are applying the same loading, here about 90% of total capacity. And we can do that because the loading speed, in the case of 2 columns configuration, is decreased. So we can see on the left part of the table, 217 centimeters per hour for 2 columns. So this is, of course, not always the case. That's specific to the example we are giving here. But this is something that we can often see in the case of the capture of antibodies. The next slide. At the bottom part, this is the result for 5 grams per liter. So at this concentration, we start only with 3 columns, 3, 4, and 5 columns, because there is no solution given by the software for 2 columns. We can't operate an Smcc process at 5 grams per liter with only 2 columns for this example, because there will be some leakage of the antibody to the breakthrough. So as a conclusion on Smcc continuous process, we are listing here the advantages of such solution. So higher loading, which allows for saving typically 50 to 90% of protein A and 30 to 40% of buffers. An increased productivity, which allows for the reduction of equipment size, which in turn allows for implementation of single-use technologies. It also can help when we want to minimize the residence time of a protein in the system because of stability issues, as Solomon mentioned in his presentation. So we know that connecting together the columns at one point and operating at high loading is going to raise some quality questions like media lifetime, viral clearance validation. But we can say now that it's clearly helping to improve the performances, and there are more and more examples showing the product quality is not impacted, as was also discussed by Solomon in his presentation. So before conclusion, just a few words about the third multicolon mode that we mentioned in the introduction slide. So what we are calling multi-step is a mode where on the same equipment, the colon from a first chromatography step is connected to the colon of the next chromatography step, which is itself connected to a third colon. So here the elution from the capture step is going to the viral inactivation, then from there going to the next ionization step in flow-through or bind dilute mode, and then the elution from the second step is being directly loaded to the third one. So there are several case studies published by Biopharma on that processes that are going to bring another level of intensification for the DSP with drastic footprint reduction, a much faster processing, and much higher productivity compared with the current setups, because of course of the whole vessels and the storage between unit separation that is going to be not required anymore. On the graph here, we are not working under continuous loading for the capture step, because the first capture here is a normal batch capture step. The first column is working exactly like a batch column. However, and this is going to be the conclusion slide. There are some process development teams that are already working on such multi-step, multicolon chromatography processes using a single equipment with the integration of the continuous capture, either batch multicolon or sequential multicolon. So there are some reports with productivity values that are increased 4 to 8-fold compared with the initial batch process one. This by performing a continuous loading under batch multicolon mode, but also integration in the same equipment in the multi-step chain together with some inline dilution and inline adjustment for buffer preparation. So as a conclusion statement, there are now several options available in terms of continuous chromatography processes to make the switch from batch to continuous DSP, to make the switch from a single colon to multi-colon approach. It's certainly not easy to make the choice between those options, but there are some modeling and simulation tools to help, and there are some proof of concept and demos equipment available to check. We want to show here that the suppliers are progressing and are proposing more advanced solutions to match the needs of the biopharmas and to address the challenges of DSP intensification. So this is going to be the last slide. Thank you. Floria, I think this is going to be the question session.
Yes, thank you so much, Yvan. Now we will begin the question and answer segment of the webinar. Once again, I would like to remind the audience that they can still send their questions via the questions tab on the right-hand side of the screen. The first question is, what are the approaches suggested for demonstrating viral clearance of the capture step? Solomon?
Thanks, Ursula, for the question. Yeah, so this, as I covered in the slide deck, this is definitely one of the considerations in terms of development for to demonstrate viral clearance, and there are some good references out there. So Biocon's approach essentially is at the first step when we're entering into continuous capture, we've enabled a methodology to actually get the system into a virus testing lab, and our intention is we have two approaches in mind. The first essentially is where we're going to break down the continuous process into two parts of testing. One is the worst case scenario of overloading, and the second case, essentially the second pass approach for the second column, where we will be testing this, not necessarily in the full process mode of 16 hours. So the key part over here is to study the process in the steady state and to evaluate whether the startup state and the end phase has any impact on the viral clearance. So essentially, this is one way of breaking up the viral clearance approach. While it's ideal to get the instrument into a viral testing facility, if that is not possible, there are suggested approaches of scaling, of having an alternative methodology of scaling down the column by testing the various aspects of overloading and second pass on a batch column itself. But our approach has been to consider evaluating the process using the equipment and scaling down the process only by column diameter.
Thank you, Solomon. Question related to the resin lifetime. Can we monitor the evolution of resin capacity over the during production? Yvan?
Yes, so the question of the resin lifetime, so the monitor like online during production can be made because we depend on the design of the equipment actually, but with the design that Novasep has developed, each column is going to be equipped with a UV detector, and so it means that after each column we can see through the UV what is going out from the column, not only during the elution of the target product, but also during any time of the process. So we can record one UV signal that will be recorded for all the cycles, and this is what we are calling the internal UV profile. So this internal UV profile, there will be one part of it corresponding to the loading step, and at the beginning we will have only the non-retent products going out. We will see that on the UV, but at one point the first column being overloaded, it is going to leak. The antibody is going to leak to the second column, and we are going to be able to see this leakage from the first column to the second column. It's not a leakage, it's not some product that is lost because it's captured by the next column, but we can see it thanks to the UV profile. And once at the beginning of the production, the resin is new, the resin is fresh, the capacity is maximum. This point where the mud is leaking is going to be at a specific time. And as long as the protein is going to be aging, the capacity will decrease, and this point, this time for the leakage is going to change, is going to be less and less and less. So we can see that we can monitor along the cycle the time where when the leakage happens, which gives a picture of the situation for the protein itself.
Thank you, Yvan. The next question is the lifetime of resins expected to be impacted due to overloading? Solomon?
Thanks, Ursula, and that's definitely a good question, sort of related to the previous one. Our experience has been during our lifetime studies, the resin lifetime and the fouling is primarily due to the CIP cycle or the lack thereof of an efficient cleaning cycle. So from a consideration point of view, we need to keep in mind, as long as we are appropriately scaling our cleaning cycles, one is from our batch to our continuous process, and then the continuous to our scale up, essentially keeping the residence time of cleaning constant. One should expect similar lifetime, and that's what we've seen, and it sort of corroborates with a lot of the references on this front. Yes, of course, the at first thought, it seems that there should be probably a lower life cycle, given that the overloading in this case would have an impact. But our experience has been that the overloading, one would really want to watch out for the leaching of the protein A, but we've never seen overloading having an impact on the life cycle. But it is definitely a consideration. So during and this manufacturer would experience during their resin lifetime studies, but there are suggestions on doing accelerated resin lifetime studies in case one needs to do it during the POC stage early in development. So that's one option to quickly assess this. But our experience has been that there's been no impact on resin lifetime during this overloading in continuous capture.
Thank you, Solomon. Can we define a batch for continuous chromatography the same way in pharma and biopharma applications? Yvan?
Yes, so in pharma applications, the continuous process, the same way in biopharma, still involves several columns. There is a continuous loading. There is a continuous elution in the case of pharma application. So we are quite close to a continuous capture process. One difference being that in the case of the capture, we don't have a continuous elution, but the eluates are collected at regular interval of times. But this is quite similar also due to the fact that in both cases the process is cyclic. We have one cycle, and we have this cycle is made of several periods, the same number of periods as the columns as we said in the presentation. So we are continuous, but they are still it's a continuous process, but cycles are clearly defined. So at the end of the day, it was the case for small molecules. The batch definition was a certain number of cycles. It could also be a certain number of periods. So a certain number of cycles and periods, which is related to a period of time. So this period of time, this number of cycles, can be related to some production constraints. Sometimes for small molecules, the production constraint was the size of the filter dryer after the equipment after evaporation. So that could be a rationale to define the batch. In the case of biopharma, it can be the volume of the fed batch bioreactor, but it can be a number of days of production or a number of hours of production in the case of perfusion, as long as this definition is going to be a certain number, an integer number of cycles or an integer number of periods.
Thank you. Before I finish the webinar, I would like to take this opportunity to thank our presenters, Solomon Alva and Yvan Ruland, for sharing their knowledge with us. I would like to remind our audience that they can view this webinar on demand by visiting biopharma-asia.com. Thank you very much, everyone. Have a great day.